I go back to this, and I say my patient has, microcephaly and seizures, and I don't know, intellectual disability. I'm not really good at typing, but it's okay. One of the things the system does is it says, oh, well, look for abnormality of the genital systems. Did you actually ask your patient to take their pants off? Because that could be relevant for a differential diagnosis. Skeletal muscle atrophy, optic nerve hypoplasia. And then it correlates with all sorts of genetic disorders, so it can actually show you the various genetic diseases that match your symptoms. If you think one of the symptoms is not really relevant, well, you can click on that and it will recompute the differential diagnosis without that symptom. So it allows you to very quickly search the space of various diseases and help you phenotype your patient right at the bedside. And it can allow for training for the next generation of medical students. It's something that allows them, they can go and search the historic records, which were entered by more senior clinicians for previous similar patients, and they can get suggestions which are maybe not as obvious to them as to their senior colleagues. One of the places where this is used is for genomic matchmaking. So if there are two patients with rare genetic diseases that are seen at different points across the world, you want to be able to connect the doctors, but the doctors are, don't even know each other. So we've and others have built systems which allow you to record the genetic variant that's interesting and the patient presentations and connect these clinicians when a match is discovered. And this is just an example of how it looks like. So it'll say, well, your patient has all these features. This is the features in the other patient, and they both have a mutation in this gene. Is this relevant? You guys decide, and if so, go ahead and write a paper about it. So just very quickly, the people who did a lot of the hard work, this is Phenotypes, the tool, and the Phenome Central team in my group, and we work closely with people at NIH and the HPO team. And one thing I wanted to mention, I'm guessing some of the audience are undergrad students, some of the best things about phenotypes, like the pedigree drawing, and actually coming up with the phenotypes name was done by undergrads. So, lots of opportunities for research in this area still ongoing. So, that's it. Wow.
Hey everyone. Great to be here. So essentially, I have to ask you guys to excuse me. I'm just getting over a cold, so I'm not going to be as sonorous as I usually am. All right. So essentially, to summarize, you're stable. You're stable. Summarize. I can't do what I do without the last two speakers' work. And so what I do is I build biomarkers for academic projects and for pharmaceutical companies. And what I try to do is I try to really figure out what a population looks like, what a population of patients really looks like without forcing issues. So let's move forward. Okay, now the clock starts. Okay, so this is one of my academic projects. And it's called Combine. This is a cross-Canada depression study. And just to talk about myself just a little bit, in addition to this project, I also do natural language processing at the Center of Addiction and Mental Health, where I get computers to learn from electronic health records and to make decisions based on them using machine learning. So just back to the Combine vision. So what we want to do here is we want to be able to understand depression. And so we're measuring many different types of variables. We have molecular data, we have imaging data, we even have clinical scale data. And what we do is we combine all of it, or we're trying to combine all of it into a database, so that we can parse what's really going on in depression, and we can make decisions as to how best to treat patients. Right? Should a patient get duloxetine? Should a patient get magnetic therapy instead? Or other various, should they meditate? Should they lose weight? You know, there's a lot of heterogeneity between, you know, underneath this disorder. Okay, so the biomarker, so let's talk about biomarkers for a bit. So simply a biomarker is any collection of data that can be turned into a model that allows you to make a decision about your patient, right? So people know about PSA, the prostate measurement. It's not a very good biomarker, but we know that if a man has prostate cancer, it's elevated. People know that LDL, increased LDL, is a biomarker for, you know, heart disease, and so forth. We also know that, I mean, recently Microsoft, it's not that recent anymore, but Microsoft actually took tweets of women who were pregnant, and they were able to predict with high accuracy which women would have postpartum depression. And so that's an example of a biomarker as well, just from looking at at the way they use words. And I've done something like that using Facebook data. I got a machine to actually learn about people through their Facebook data, and I developed these like interest profiles, and you can actually start parsing people very accurately, and you're going to notice this happening on your feeds, okay, if you haven't already. Okay, so what we're trying to do is we're trying to divide up the population. We're trying to understand what's really going on in a disorder. Okay, but biomarker research has faced a lot of challenges, not only because it's been difficult to collect really good, accurate data, but it's also because, I mean, it's a tough job to be able to predict. We all know this. I mean, weather's still a challenge, right? And it's a very complex system. But so is what's going on behind a lot of these disorders. And I'll give you an example as we move on. And, you know, this is some of the math stuff. You know, I'm a mathematician by training, and the way I see what's going on here is there's a lot of substructure that the machinery that we're using is kind of forcing divisions where there aren't really any divisions. Or it's not seeing how heterogeneous things are, because it can't see the relationships. Okay, so machine learning. So I'm gonna answer this question, what is machine learning, with an example. I'm gonna show you guys a robot that's learning to walk, okay? So here's this video. Okay, so what's happening here is these guys put together this robot, and the robot doesn't even know what it is. It only has one motivation, to locomote. And all it does is it flip-flops around here. It's trying to figure out what it is. It's moving its parts. Do I have 4 limbs? Do I have 6? It doesn't know, and it starts figuring out that it's this thing. That it's this, I have 4 legs. Okay, now, it only has one motivation. And we don't tell it that it has to walk or anything. It just says, your center of your body has to locomote from here to here. And if you do something that translates into locomotion, then we give an award for that, and we penalize everything else. That's it. And this is what ends up happening. The machine, and this is what happens to babies. I mean, you've seen all, babies jiggle around, and they just want to kind of get going. Now, they wanted it to get up and actually start walking around like a spider, like some creepy thing, but this is actually what it really looked like here. It kind of is this kind of fumbly, right? Okay, so, yeah, so that, it worked. It figured itself out, and it started moving. Okay, so what we're doing in medicine now is we're trying to use tools that allow things like this to happen, to actually start really parsing what's going on in heterogeneous disorders. Okay, good boy. Okay, I was going to talk to you about robots learning to lie, but the video worked, and if you guys have questions, I mean, just quickly, there's been experiments in this robot evolution studies, where the robots actually learn to deceive each other, right? It's pretty amazing. We can talk, I'm limited in time here, so I was going to do one or the other. Okay, so back to medicine, okay? So the protocol in medicine is very basic. You have a bunch of patients, you collect data, and you do your best at doing this, and then what you do is you put together a training set. This is what people always talk about in biomarker research. You have a training set, okay? So I developed a machine learning methodology, and I gave it patients. So I don't know how well you can see this. I know it's difficult to see, but let me just speak loudly. So what this is a map. It's a patient map. It's something that I've created, and all it does is it takes high dimensional spaces, so lots of variables, and in this case it's just a bunch of genes. So you're looking here at about the interaction of about 11 genes. So the closer two people are on the map, the closer they're related according to that set of variables. That's it. Very easy. So a doctor would come in and say, oh, George and Bob Oh, look at that, from this perspective, they're really related according to this perspective. And what you've seen here is the machine learned how to separate the blues from the reds. So the reds are an adenocarcinoma and the blues are squamous cell carcinoma, right? So it's cancer that you want to treat differently. And what's happened is it learned that, but now the real test, and this is the real test of biomarkers, is if I throw a brand new person inside, did it actually learn anything, like that machine learned how to walk? And in this case, it did, right? It actually did. If you notice here, each one of these is a new individual that gets put in, and it does very well. It actually recognizes the squamous cell and the adeno quite well. It makes some mistakes, and actually 2 of them weren't mistakes according to surgeons. It was mislabeled, so the machine was smarter than I am, right? It actually was able to see it. So what you're actually seeing here, and this is what I want to teach you how to read. These are these patient maps I've developed. They're 2-dimensional, very simple to read. The closer people are on these maps, the more closely related they are according to the variables you put in, whether they're the variables, language-based variables or variables that you pick up from devices. Okay, so here's an example with microRNA. Okay, so there's this very commonly used machine learning technique called support vector machines. Okay, they're very good. They're used everywhere, and you probably even inadvertently used it for some of your online stuff. And so what we did is we wanted to see if this thing could learn about depression. And what ended up happening, so here's the data. We had about almost 3,000 microRNAs. MicroRNAs are just these molecules. You guys probably don't know this, but those of you who don't, they're part of our molecular circuitry at a very deep level. And so we had responders and we had remitters, and we're trying to get the machine to learn. And what it did is it did this. Okay, it figured out some microRNA. I can't put them up there because we haven't published it yet. And what it did is it basically, with 72% accuracy, it did its best to figure out how to separate people who went into remission versus people who didn't when they were given a drug, specifically this duloxetine. Okay, but then I have a different philosophy. I say, okay, look, let's not force a model. Let's not do that. Let's actually really try to figure out if there is a very pure group, according to the variables you put in, right? Just think about this. If you're measuring one variable, how likely is it that you're really going to be able to capture what's really going on in a disease? Do you think you can actually parse depression with one variable? It's very unlikely. Okay, so what I decided to do is say, okay, look, we have to accept that sometimes we just don't know. And this is what ends up happening. I built a map on this, and what you end up getting is you get a region where you can't make a lot of decisions. You just have to say, okay, I don't know. But it found a very pure region where it's really recommended that you do not give this medicine to these people. And look, it's very, very accurate. It's very pure, and it reproduces. So suddenly you have the ability to make a clinical decision with extremely high accuracy. And then what you can do is you can throw other variables in. So the point here is that you're no longer forcing the computer to learn how to walk by just using one type of movement. You're saying, okay, this one type of variable that you're using is not capturing the heterogeneity that's going on, but it's capturing a part of it. And so this is the microRNA. So okay, and here's another example, and there's something going on in the corner, but then you get these guaranteed responder kind of group. It finds these predictive islands, as we call them. And this is where we're moving towards in the whole biomarker business. And what ends up happening is you get a lot more you have the potential to develop way more accurate biomarkers, and you actually end up helping clinicians finally really make decisions. Okay, so what ends up happening here is what you can do now is you can do this repeatedly. You can say, okay, and this is a horrible, I hate this image, but you'll get the point. You do it again. You put another type of variable. You use some data that Kai picked up from a device. You throw it in, suddenly you can parse out a whole other subpopulation. And you do this over and over, and you really start understanding that depression, in this case, has an inflammation component, it has an apoptotic component, it has some neurological components, obviously, but also has lifestyle components. right? And Kai's technology would be really great at picking up things like this and also linking it to other physiological components. And now we're starting to actually try to, integrating. And that's basically my story for now, and thank you.
Okay, well those three talks were fascinating. Thank you very much. So the floor is open for questions. I just want to check. How much time do we have?
Like 25 minutes, yeah.
Oh, very well. Okay, great. So lots of time for questions. So I want to get the discussion going right away. Any questions hot off the press from you? Okay, then I'm going to take the moderator's prerogative and ask the question. So I was struck by a couple of things. One was, Kai, you talked about the kind of devices, for example, figuring out how long you've been in a bad mood for, which I'd love to attach that to my teenage daughters, because I really have to understand that. But I'm wondering whether that is an example of the kind of variable that could be added to the models you're creating, which would then be able to predict response to medication potentially. Is that am I imagining that, or is that an example that makes sense?
Well, so this is something that I'm already trying to do, right? But the problem is the data has to be of extremely high quality, which is why we rely on people who develop, Kai's type of technology. But that's exactly it. You integrate this along with, like you throw it into the mix, right? So what we know genetic markers, and it's sometimes things are happening because we're driving them to happen through our behaviors. And sometimes our behaviors are indicative of physiological, genetic things that we have. Like, the breathing patterns, I'm sure, are one simple example. Yep, you got it.
I think I love all the three presentations. It was very interesting. But I have a question. Probably I'll add to Kai 2 questions. Are those data going to be useful? And then where are we going to use those data? Probably the problem with the big data, if we don't know our in questions, we can go any direction. And there's no way to figure out what question we're answering first. So the important to know which we have a very complex health system. When you collect those data, what questions is going to answer? What is going to be, when you get those answer, where are you going to implement? Or how are we trying to figure out the system which can going to work? And the second speaker talks about probably is collecting millions data probably not work. This might be collecting the useful information. That also I was trying to say again, if we collect it from only disease population data, we know only one set of population. We're left out all the other population, and then it's again another challenging. How do we bring the balance, understanding of the disease population and understanding the people without disease, and some sort of come out the.
So we put a time limit on the question here.
Okay.
So
First,
I think we had two questions. One Kai, and the second one was Mike.
So I agree with your point about needing questions framed in order to structure what it is that we're collecting. My approach is more about looking at creating these general purpose data collection approaches or mechanisms, not looking at the questions that people want to answer or the specific kinds of applications. So the example that I gave with respect to looking at the noises that are occurring inside of our throats, for that particular project, I just looked at it as can we do the sensing without necessarily looking at what that sensing will help answer. And then I pointed to 2 projects, right? So one at Cornell where they're looking at breathing behavior, they're also looking at sleeping patterns out of that. The second is out of UCLA where they're trying to track food intake, right? And so what I believe is there's going to be a lot of kinds of questions that we want to answer, but we won't be able to unless we capture data in such a way this flexible manner that allows for us to process and extract meaning from later.
I guess to follow on, I think Kai had this great dichotomy in the data that, broad data that we will capture, and then we'll figure out what to do with it later. And, the more, data that can be captured by integrating the capture into the user interface, but then having a very sort of specific data that can be captured. I think our work falls more into the second class, where we're customizing the user interface so that the clinician can capture the relevant data while also going through the rest of their clinical workflow. To answer your question about the capturing data just from the disease cohorts, I mean, for a tool like for what we're doing, we're actually capturing data for every single patient who comes into the door of the genetics clinic. Obviously, this is a very biased sample set, but there are also population norms that can be integrated into the tool. One of the things I didn't show is that if you enter the patient's head circumference into the tool, it'll right away tell you, is that normal, is that abnormal, is it, where does it grow on the growth chart? If you're having multiple measurements over time, you can track that over time. So all of that needs to be integrated, and obviously population norms are critical in being able to capture data effectively.
Yeah, and just to add, I mean, I think you're bringing up a very good point, and it's been addressed, but I just want to reiterate. I mean, and to structure it, there should be projects where we take this data and we examine it for the way the population breaks up, looking at these different perspectives, right? This whole perspective analytics that I end up talking about all the time. So you want to see it. And at the same time, you have clinicians and scientists who are not necessarily mathematicians who say, okay, you know what, this particular aspect from these two collections, let's include these and let's do an exploration. And what ends up happening is you generate hypotheses, real science, and then you test them, right? Don't believe what your models tell you until you repeat. And then either if it's just in general or for a very specific disorder.
There's a question over here.
Yeah, great talks. Two speakers mentioned text mining, and I know a little bit. My perspective is, I'm wondering if we should just wait for someone like Google. So Google has the most amount of books in the world that they've scanned, and they're working on understanding language. When they get to some level of understanding language, could we bootstrap that to read, say, electronic medical records that a doctor wrote? Would that be a good? I guess that strategy sort of wait, but
We're doing it already.
okay.
We're already actually using these books. Google's already put this data out. You can download it.
Yeah, it's, I mean, we're doing it. The question is, what is the accuracy of the result?
Yeah.
So, for example, certain things, like if you wanted to predict from electronic health record whether a patient has diabetes, it's actually really, really easy. If they've ever been prescribed a set of drugs that includes insulin, they have diabetes, and if not, probably no. It's sort of, that one is very, very simple. If you wanted to predict if they have dementia, it's very difficult because of the differences in the terminologies that people will use. We do use the text of the electronic health records for various mining of the terms to give them as suggestions to the clinician so that they can say, yes, that's relevant, or no, that's not relevant. Similarly, we use automated translation because the dictionaries the definitions, the terms have been translated across languages, but the definitions of the terms have not. And for our search algorithms, we need translations of the definitions to search across. So we use automated translations of of the definitions to search against, but we don't show them the user the translation because that actually is pretty poor quality. But at least we get the words in there, even if they're in the wrong order.
One of the big challenges in the application is the expectation that physicians in their offices can devote sufficient time to the kinds of questionnaires to produce an accurate output. Given that currently patients and clients going to physicians resent the fact that their doctor is looking in a computer screen more than at them, how can you reconcile that in terms of effective care in the community? Because the reputation that's now very prevalent, of course, is that current graduates in med school do not have the same expected personal interaction with their patients as in the past, for the sake of regularizing a process. That's point number one. Point number two, what questions are being asked by these various creations of documentation that's super perfect, that actually asking 1,200 variables? What precise questions are being asked for answers? Or are they being done in the hope that it will reveal something in the future if that information is being interrogated?
So I guess let me handle those questions in reverse order. For in terms of which questions are being asked, the questions that we ask have to be targeted based on the answers that have already been provided. So it's like a game of 20 questions, right? Where you ask a question and then the answer is yes, no, and based on that, the whole set of questions which follows have to be targeted to what has already been asked. So, if somebody says, in the game of 20 questions, if somebody, you try to guess an object based on 20 yes/no questions, if somebody says it's inanimate, it's not useful to ask, is it a dog? Because that's false. So similarly here, you ask first very broad questions, and then you can target, the system should be able to target based on what other things could be relevant for the set of diseases that match the already selected phenotypes. And so it has to be dynamic and has to be learned from the data using machine learning and targeted to the individual patient record. For the second question about having a computer out so that I can look at the screen and not at the patient, it's a big issue. There's a lot of work around how to make the same process more transparent. For example, if you give the clinician an iPad on which they can write with a stylus, is that any different really than them writing on a piece of paper with a pen? Is that going to be less interactive? What if we actually mount a camera on the clinician so they can video record every single thing that they see? Is that going to be more intrusive because they'll be recording you, without your underwear in your full glory? Or is it going to be actually better because it's going to be giving them an accurate record of everything that was observed and not just the things that they were able to take down during their interaction? These are very hard unanswered questions that a lot of researchers in the field of human-computer interaction are looking at right now.
I mean, I want to add, it's a great question, I want to address how medicine has to change to adapt to participatory medicine. I was a chief science officer of a company in Boston, and we interacted with women, and we were just focused on women. And one of these diseases we were focused on was polycystic ovarian syndrome, and women are really pissed off. I mean, they have 18 seconds with the doctor, and what ended up happening is the CEO had this image that that patients would actually participate in their own care. They do their own research. They're going to have tools. Eventually, things like Michael's tools are going to become part of a medical AI, that you're going to be able to go online. It's going to very quickly get you near where you are, and it's going to make recommendations. And the medical doctor, the new medical doctor, is going to be this frontline soldier in this medical science, which is going to be to interact with the patient. And maybe there has to be, there might have to be multiple sessions, and one session is going to be, what do you think? And they interact. It's not going to be this authoritarian relationship anymore. I think something like this is going to have to switch.
Terence?
It's a question about the depression and microarray data. So I understand when you get to the point you've got support vector machines and you're able to classify the two groups, but two questions, maybe because I'm a statistician. Support vectors don't put probabilities on things.
Right.
So there is a gray zone in the middle, so how do you deal with that gray zone?
Yeah, okay.
Second question,
Sorry.
Is how do you deal with the piece before you put it into your support vector machine?
How do we deal with, sorry?
How do you deal with the processing piece before you throw it into a support vector machine? So if you've got billions of SNPs or microarray data, isn't there some kind of data reduction technique? Like you had a nice 2D plot. Well, how did you get from billions of SNPs to.
Okay, so you and I should chat later, but I'll answer this quickly. Okay, so number one, I have removed myself from p-values completely. Okay, so what I work with now is I have a model and it fails. And I'm like, this is crap, right? And then model, and it's rigorous, like I'm a physicist, right? We have a theory, we test it, it fails, done, right? So what happens is when things replicate locally, then we attempt to replicate outside. And you can use, I don't care about support vector machines. It's one of the things I have expertise in, but there's 100 other things I use as well. But you get, so I think that answers that question. The paradigm I'm using now is a little bit hard-assed because I don't believe in cross-validation either. As a statistician, I know you know what that means, right? Bootstrapping, cross-validation, I don't believe in any of it anymore because I've seen it all fail over and over. I believe in an independent data set, and then I'm still like, right? We have to do it again. And that, because biomarkers, this is very, I mean, a clinician has to use this to make a very serious decision in many cases. Okay. Now, the 2D map that you saw, that's a technology that I created that translates the high-dimensional space into a map. So don't think of it as numbers. It translates the relative positions of patients on a 2D map, and the way it does it is I accidentally bumped into an old friend called the discrete dynamical system, and one day I realized this thing has memory. It's able to translate thousands of variables into a geometry. Why can't I use this, et cetera, et cetera, et cetera? And then I published a paper on this where I did some cancer research, and I'd be happy to talk to you about it. And now I have a new technology which is way beefier, but it's commercial. So,
What magnitude of data are you looking at if you're trying to look at like supervised learning for machine learning to make those test databases?
you're asking me how many patients?
Or patients or data points from like independent variables, yeah.
So how many what would I like to see, or how many am I dealing with in real life?
How much do you like to see?
10,000 patients.
Okay.
I mean, after doing this, have you guys heard of D-Wave? They're a Canadian company trying to build a quantum computer.
Yeah.
They fly me over them. They're like, we want to talk to you about cancer and quantum mechanics. I'm like, okay. Right? He's like, you're one of 2 people on the planet who have both. I'm like, okay, let's talk about it. They didn't want to hear about any of this stuff because they wanted to see 10, 20,000 data points. And I'm like, that's not realistic for our clinicians. So what I'd like to see, I agree with them part way, but in real life what we get is 200, 300. Like when I do work for pharma companies, I get 1,000 people. I'm like a kid on Christmas morning. Like, wow, this is amazing. Right? But yeah, I mean, I don't think you're going to capture, I mean, that lung cancer, that was only 104 patients, and it's very accurate. So sometimes, and that's the tempered side. Sometimes you don't need it. Sometimes things are simple. Sometimes a few genes really will allow you to separate. Sometimes a few brain areas in MRI will allow you to say, yeah, there's something real, but 10,000.
Great talks, thank you. I was wondering, I actually would like to get your opinions on the application of the machine learning techniques on the probabilistic record linkage and where we are standing on now. Is it mature enough to be replaced with, I mean, the traditional probabilistic record linkage to be replaced with the machine learning techniques?
Well, I mean, there's work to be done. And I think we need to be sober, and that we need to test. But I think some of I mean, you guys understand that some of these deep learning work that's been done at U of T with Hinton and company, great scientists, and he's now been absorbed by Google, right? He's still a professor here, right? But he does that as well. I mean, these are they're very good at a few things. They're really, like, it's starting to become very impressive. They can recognize any species of dog with high probability, but this is not what you're talking about. You're talking about actually like electronic health records, right?
In general, actually, at ISIS, we try to link different databases across different ministries, and just to be able to trace individuals.
The quick answer is that if you don't invest in this now, you're going to be left behind. I mean, that's the truth. Yeah, machine learning definitely has a place, in my opinion, but it has to be done very rigorously. I spoke to ISIS just 2 months ago about some of the data they're collecting, and they're like, yeah, actually would be interested in talking to you in a year. And then last year they said that too, right? So I'll bug you again.
Yeah, I just kind of wanted to jump off of the point that you made about patients becoming more involved in their diagnoses and their outcomes. So I am starting to think that there's going to be an increasing need of literacy on this subject and this topic, and I was wondering if you had any ideas of how to make stuff like machine learning and how it impacts medicine and potentially other fields as well to the general public, because eventually at the end of the day this is data that's coming from 7 billion people on the planet. Well, hopefully, ideally, if we can get that kind of data, that'd be amazing. But, you know, this is all data that's coming from people at the end of the day. And, you know, just sitting down and even talking about some of this stuff in a room of very highly trained people can be tricky at times, let alone for the general public. So how would you kind of demystify the space around this topic so it's accessible to everybody? Do you think we can get there within the next generation or so?
This is an excellent point. I mean, education is a vital part of, you know, this challenge I put to the new clinicians. You know, and I consider myself part of this new challenge. And I mean, I think really a big part of the education has to do with science literacy. Besides, and before, I don't know, Neil deGrasse Tyson is doing a good job. I mean, in all honesty, look, in the next 10 years, 3 billion people are going to come online. I mean, are you guys aware that Facebook is preparing drones right now? I mean, this is not BS. Google is going to have satellites in orbit shortly in 10 years. It's going to provide internet for everybody in the world. The amount